greatest minds said that you'll never be able to form a molecule in space because space is too empty. Space is too harsh. You can't, it's going to just break apart molecules, it's going to break apart bonds. Now we find molecules routinely.
We've probably seen all the great pictures from the Hubble Space Telescope. What they do is they look at a particular color, a yellow or a green and a blue, and then they combine all the colors together to make a picture. Well, in ALMA, we could do better than that. What we could do is we could actually see shades of yellow. We could actually see shades of red. And it's this shading that tells us what the universe is made out of in molecules.
What we could do is go to a laboratory, like here at the University of Virginia, and actually put molecules in a large bottle or a large cell. And what we do then is we excite the molecules and actually measure the colors that come off of those molecules. After that, we take those colors and directly compare it to the data that we get from space to see if those molecules are actually present in space. So what you see in front of you, all these little bumps and wiggles, these represent the shades of color that I was just talking about. We know exactly what this molecule is because we put that in our laboratory cell, so we know what molecule we're looking at, and we were able to identify that molecule by all these little bumps and wiggles. We then took ALMA and pointed in the direction of Orion, and we're able to take some data there. And when you put the laboratory data right on top of the data from ALMA, you see an exact match. That is exactly how we know that this molecule is sitting in the Orion cloud over 1,000 light years away. And it's basically the sensitivity of ALMA that allows us to do that. It is so sensitive, it can actually see the traces of these molecules in these clouds that are over 1,000 light years away.
How common then is the chemistry on the Earth found in the galaxy? There was always some idea that the organic chemistry, the molecules that contain carbon, nitrogen, and oxygen, the Earth had to be very, very special in order to drive this organic chemistry to form big molecules, big organic molecules like RNA, DNA, proteins, amino acids. Not so much. The universe is doing this naturally. It's forming these molecules naturally in these very harsh environments of space. And this is what we're able to find out with these observations that we did with ALMA.
The search for alien life has begun.
We think that other worlds, perhaps other Earths, if you will, exist. We know of hundreds of other planetary systems right now, and that may be the tip of the iceberg.
It isn't conceivable, really, that there would not be life. Is it intelligent? I don't know. That's a whole other issue.
Within a few decades, unmanned probes equipped with artificial intelligence may find life on planets beyond our solar system.
It's certainly true that life needs liquid water, but it only needs liquid water when it's growing or reproducing. When organisms are dormant, they can dry up and be in a dormant state, and we don't know what the limit is on how long they can be dormant. For all intents and purposes, some organisms can be dormant forever. So you can imagine a dormant bug inside of a rock being kicked out into space, flying for millions of years even, landing on another world, that rock breaking open, and that bug getting wet, getting rained on, coming back to life and crawling out. One giant step for a small bug, one small step for a bug, one giant step for bug kind, and here they are on Mars, or that could have been Earth, and the bug could have been flying in from Mars. So the ability of organisms to spread in the dormant phase is something we don't even fully understand. And my thinking is that it's entirely possible for organisms to spread from planet to planet, and I think even from star to star, because they can be so resilient in the dormant phase. They're not growing, but they're alive. This is, in a sense, what we've done to Mars. We've put organisms inside spacecraft on Mars. They're not growing. They're not doing anything. They're just sitting there. They are a potential to grow if conditions ever changed. And this is a very important distinction. Life needs water to grow, but life can be dormant almost forever, just in dry, dehydrated state, living inside rocks, dormant inside rocks for a very, very long period of time.
I think Enceladus is probably the best bet at this point from, the new news I'm hearing. Primarily because it's spouting the water out at you, you just need to go collect it. You don't need to drill hundreds of kilometers of ice. We have a very strong case for a habitable zone at the Phoenix landing site on Mars, and strong enough that we should go with a life detection mission and determine if there's life at that site.
One could look at the electrified plants. Here's this thing that looks like a plant, and yet it's zapping things out of the sky, and you sort of wonder, what is it doing with the things that it zaps? Is it simply frightening them off? Is it killing them and eventually eating them, and we're not seeing that? And so it gets back to the question of, is this a plant or is this an animal? But the plant and animal distinction is one that's very peculiar to the way that humans look at life, because we live on the surface of Earth, and as it turns out, at this stage in the Earth's history, there's only two big lineages of living organisms in the land. The plants, they're green, they're photosynthetic. The animals, they run around, they eat things. And so we see that as a very important distinction. It shouldn't be a surprise to us at all to find an organism on Darwin 4 or any place else that combines aspects of what we might think are plant and animal.
So here comes the virus, and it's going to land on one of this guy's throat cells. So notice it's covered with little yellow knobby things. You call these keys, right?
Those are the keys.
Yeah. This is a key, this is a key, this is a key. Okay, if the keys on this virus happen to fit the locks, which are those little purpley stick-uppy things on the surface of the cell, if there is a match, the cell, watch this, welcomes the virus in. And what's this?
This is the welcoming committee. They all interlock with each other, and they pull this membrane down into the cell.
And down it goes deeper and deeper, and that welcoming structure disperses, and the virus capsule bursts, and out comes the secret recipe for how to make more viruses in those little noodly things. So this unsuspecting cell has been tricked into guiding these virus recipes right into its own command center, the nucleus. So in they go.
And they are immediately recognized by this big pink molecule, which is a mini factory.
Yeah, what is it doing?
It threads the nuclear material, the instruction code of the virus, through one hole, and out another hole comes a brand new instruction set.
So it's a copying machine making copy after copy after copy of virus recipes, which then go out of the nucleus to little chefs, those blue peanutty things. They cook up proteins that go back into the nucleus, where they are reassembled into baby viruses, and then out they go. They get covered up and head to the surface, where they get new keys, and then boom! Here they come. This is an eruption of virus after virus after virus.
Let me take you through a little video now of how the virus chip works. So again, it's a solid substrate of glass, and on that glass we've printed or deposited lots of DNA in little teeny spots. So there's the little spots. Obviously not to scale. Some of these spots might represent SARS, for example, just highlighting them here. Some of those spots might represent sequences from dengue. There's no order to the spots on the array. And if we zoom in, obviously not to scale, you can see little DNA molecules stuck to the glass. And these were deposited using robotic printers of our design and production in my lab. Now, a sample is applied to the chip. Come take a fantastic voyage with me now, as we then drop onto this chip sort of Roadrunner style. And this sequence, yeah, you know what's going to happen. These sequences that are labeled with fluorescent dyes will then be allowed to hybridize to the sequences that are on there. So here's this complex mixture of nucleic acid. A lot of it's not virus, a lot of it's host, only some of it's virus, and this is a virus spot. And one sequence finds its match. The complementary base pairs, hybridization occurs, and by virtue of hydrogen bonding and energy, that sequence stays there. Again, another sequence touches down, and another, and so on. Finally, the last step of this process is to just wash away all this material that doesn't belong. And we're left with the good stuff, the fluorescent label stuff. And then a giant laser beam, it's really small actually, but it looks giant here, comes zooming by and illuminates the fluorescent dyes that are stuck to this nucleic acid. And that allows us to visualize which spots had hybridized material to them. And so we zoom out for the array, what we should see is some complex pattern of spots that are lighting up with fluorescent dyes that indicate what was in the sample. Now, you're going to have an opportunity, for those of you that signed up in the bioinformatics exercise, to take data from real patient samples of actually infected individuals and analyze the raw data from these chips. And your goal in that bioinformatic exercise is to tell me what virus was afflicting those individuals. And it's going to be exactly what you do if you were a graduate student in my lab. I'm not going to pull any punches.